Hello and welcome. I'm Aurelia,
I'm JoJo.
and we are presenting our semester-long project for gene and genome analysis.
So using multiple experimental methodologies, we studied genomic functionality using the model species Brachypodium distachyon. Brachy is an easily grown species with a relatively small genome and a short generation time, but research surrounding gene function is limited. Brady1G67840, or as we called it, the MAC gene, was our gene of interest for the semester. With further research, we could reach beyond the laboratory and use it to improve crop production by studying how certain genes function in the immune response when infected by a pathogen. Although we didn't look into it, we considered that there could be minor health benefits using the MAC gene to understand the function of the human homologue.
We began the experiment by planting 7 wild-type and 14 mutant Brachypodium distachyon plants and randomizing them. Various experimental bioinformatic and statistical methods were applied to attempt to determine the function of the MAC gene. We started by extracting the DNA from young B. distachyon. We then used the sequence derived from the extraction to design left and right primers in order to run a PCR. Gel electrophoresis and spectrophotometry were then used to quantify the PCR products, and once the bands were bright enough to be observed, we were able to extract and purify the DNA for sequencing. Sanger sequence analysis helped us classify our mutant and wild-type samples based on the presence of a single nucleotide polymorphism. Immunohistochemistry gave us a visual understanding of potential physiological effects on the structure of the stem. Statistics and bioinformatics were used to support results produced in the wet lab experiments. Initially, we needed to develop an F-Ganesh model to develop the working gene model that became a key part of our research throughout the semester. With our sequence in the models, we were able to find connections between brachy and several other species. Literature was used to hypothesize an expected gene function based on experiments performed in other organisms with a gene of similar structure. By blasting our sequence against those of 4 relatives to brachy, we were able to create a phylogenetic tree outlining evolutionary relationships between the MAC gene and other genes in brachy and genes in 4 other species. From there, we analyzed expression levels using an EFP browser as well as data provided to us. Bringing everything together, we were able to propose an experiment to test genomic functionality of the MAC gene.
Using the NCBI standard nucleotide and protein blasts, we were able to identify the MAC gene as Brady1G67840 due to its location within the brachy genome. Our initial experiments were designed to study a mutation within our gene that has substantial impact on gene function. The mutation we chose to study was NAN184, a single nucleotide polymorphism that modifies the tryptophan codon into a stop codon. The sequence shown here is the protein sequence generated from our genomic sequence. Highlighted in purple is the region of the protein sequence where the premature stop codon occurs. The other highlighted regions can be ignored for now, as they represent something that will be discussed in later slides.
The MAC gene model was formed using data derived from Phytosome, F-Ganesh, and NCBI. The original sequence is shown in teal at the top and contains the unknown mutation. From this sequence, the blast provided similar transcripts, with the most similar transcript being the TAN. The predicted sequence coded for a BDI-BD213. Lastly, RNA sequence expression can be seen at the bottom. While levels of RNA expression are not as high as expected, they clearly show the markings of introns and exons present within the sequence. The red arrow here points to the rough location of the mutation, NAN184. While there were other mutations that were located with the blast, shown by the yellow arrows, the mutation highlighted correlates to the most significant effect, the creation of a premature stop codon. It is the mutation that was chosen to be studied throughout the rest of the semester. This gene model was used to find relatives to the MAC gene in order to further study the function of the NAN184 mutation.
From NCBI blasts and research, we were able to develop the gene structure model at the top of the slide. This model shows each of the domains that were found within the MAC gene: CC, NBS, ARC, LRR. Research shows that the CC domain is linked to the activation of cell death. The NBS domain is responsible for nucleotide binding, and the LRR domain functions to recognize effector molecules. We couldn't find much information about the function of the ARC domain, but it could be worth studying in future experiments. Directly below is a model showing the function of the LRR domain, which we felt was useful in understanding how the MAC protein may function within a cell. The 2 protein bodies embedded in the membrane on the left would be the model for the MAC gene based on the presence of the LRR domain. The MAC gene is an R gene, or a resistance gene, that encodes a protein that localizes to the membrane. The figure represents how the protein interacts with G protein coupled receptors, or GPCRs, to elicit the hypersensitive response in plants.
Here we have two ribbon models of proteins that are similar to the MAC gene and that they share some of the same domains. In CED4 on the left is the C. elegans homologue, and APAF1 is the human homologue. Both are important for regulation of apoptosis, or cell death, in their respective species. This image serves to model similar proteins in order to visualize the potential overall structure of the MAC protein. Highlighted are the domains present.
In our literature search, we found two papers examining genes in other species that helped us mold our hypothesis regarding the function of the MAC gene. In this figure derived from a paper by N. Hatsugai, it was found that RPS2, an R gene similar to MAC, was important for the hypersensitive response in Arabidopsis. In his experiment, Hatsugai showed that RPS2 was highly regulated by AP2M. A mutated form of AP2M resulted in decreased RPS2 function in disease response, supporting our hypothesis that MAC may play a key role in disease resistance as well.
The next study by Shiwai Bai examined the functionality of MLA10 in tobacco and barley. MLA10, like MAC, contains the CC, NBS, ARC, LRR domains. This led us to believe that MAC likely functions similarly to MLA10. An interesting point that Bai highlighted is that the CC domain alone was sufficient to elicit a cell death response.
To further understand genes similar to the MAC gene, we performed an NCBI standard protein blast comparing MAC to genes found in Arabidopsis, Brachypodium, Oryza sativa, Amborella trichopoda, and Solanum lycopyrruscum. From the tree, we can see that the MAC protein is strongly related to various proteins found in O. sativa. Other proteins included in the tree are pertinent to disease resistance. We hypothesize that the MAC protein is also involved in disease resistance.
Bringing all the information together, we hypothesized that the BRADY1 G67840 was an R gene within brachy that is responsible for regulating the cell death in response to immunocompromised tissue. The hypersensitive response is a feature found in plants that occurs when exposed to a pathogen. The cells surrounding the infected tissue are triggered to die in order to contain the spread of infection. Additionally, we predicted that the BRADY1 G67840 was responsible for regulation of the xylem. Within plants, the xylem is made up of two types of dead cells, tracheids and vessels. However, only vessels are present in Arabidopsis. In the center, to allow for water to pass. Since the domains of MAC gene regulate for cell death, it was hypothesized the cell death led to formation of the xylem.
Histological analysis showed that the mutant samples were irregular in structure and had noticeable changes in their interfascicular fibers when compared to wild-type samples. Mutant interfascicular fibers appeared thicker with necrotic areas embedded within. Measurements of wall thickness were averaged, and a t-test was performed to determine if there was a significant difference between wild-type and mutants. The thickness of the metaxylem was also measured, and a t-test was also performed on the gathered data.
Quantitative analysis of our data was performed and is represented by the box of whisker plots here. From the graphs, we can see that there wasn't much of a difference between mutant and wild-type metaxylem. However, mutant intervesicular fibers were almost double the thickness of wild-type samples. This shows that the mutation caused an increase in vesicular fiber thickness and not xylem, like predicted by the hypothesis. These are the t-tests that support that data.
The heat map shows the highest expression values in young leaves during a normal thermoregulation cycle. We expect expression levels to be high in leaves because leaves are more likely to become infected by a pathogen, and eliciting a cell death response in the leaves is less costly to the overall health of the plant. As appendages, leaves can fall off and die without causing harm to the main vascular system. Once the stem is infected, there could be repercussions that result in total plant death.
Here's the graphical representation of the heat map levels of expression, with the young leaf being the highest level of expression. Here are the t-tests that support that data.
The seeds provided at the beginning of the semester were assumed to be correctly labeled by those who studied the gene in past semesters. The expected genotype was heterozygous mutant with a single nucleotide polymorphism change from a G to an A. Our data derived from our extraction showed something different, though.
Figure 9 shows the Sanger sequence and sequence alignment of the pre-spring 2020 data. The highlighted region in the sequence shows the mismatched TGA and TGG codons. This is indicative of a true mutant and is supported by the sequence alignment to the right that shows a double peak at the mutation location.
In figure 10, the Mack-Sanger sequence shows a homozygous TGG that is supported by the single peak found at the mutation site in the alignment to the right. There are many factors that could have possibly contributed to the lack of a true mutant. However, we came to the conclusion that it was likely down to seed handling prior to planting.
On our analysis of Sanger sequence from our mutant 1 and 11 plants, we discovered that our mutants were not the expected heterozygous mutant, but rather a homozygous wild type. Pre-spring 2020 data did have a true mutant showing a TGA in place of TGG. It is suspected that the seeds provided were not true mutants and the package must have been mislabeled.
The proposed experiment was created to determine the effects of the mutation. Since the original mutant species used did not have the NAN184 mutation, a known true mutant must be identified and used instead. In order to test the regulation of the mutation, a GFP project will be used. The seeds will be mutated to insert a GFP gene. From there, in order to determine true mutants, DNA extraction and PCR will be performed. Once true mutants are determined, each species would be exposed to Puccinia brachypodi, or leaf rust, which attacks Brachypodium distachyon and its relatives. Over the course of exposure, we will measure expression patterns using GFP tag species, with particular focus on younger leaves where the gene expression was seen at higher levels. To measure the alive to dead cell ratio, we will measure electrolyte leakage, which is a common method of measuring cell death. Sections would also be used to measure the thickness of interfascicular fibers in mutant and wild types. This slide provides a slightly more detailed explanation of that experiment.
We hypothesize that mutants will have a weakened response to infection. This would be demonstrated by an increase in the spread of disease to surrounding tissue or a non-functional hypersensitive response. We also expect to see an increase in stem rigidity due to the thickening of interfascicular fibers. Here are references, and we want to thank Professor Hazen and Katie Webster and Katie Dorfman for all of their help throughout the semester, both in person and online.
Thank you.